Thank you very much, Wanjen, to coordinate these sessions and the time change also, the European time change. We agreed that I should say a few words now about the three presenters because today's topic will be presented by three young scientists, all from Beijing Forest University. It's Professor Zhang Chunyu, Assistant Professor Fan Chunyu, and doctoral student Hao Minhui. They are three young scientists who have published widely in leading international journals. And I'm very, very happy that they agreed to address us today about the topic, which is beta diversity, a very important topic. But a bit complicated. I worked a bit with all three of them, and I found this extremely challenging, beta diversity. Well, Professor Zhang Chunyu is one of the top scientists, the top young scientist in his country, and the other two also are very, very young, energetic scientists, and it was a great pleasure to work with all three of you. Well, Chunyu, would you like to start?
Yes. Okay, thank you, Gado, for giving us this chance to introduce our research work. Today's topic includes three parts. Part one, I will introduce how to calculate the Null model for beta diversity. My two young colleagues will join me in today's discussion. They have done very excellent work on the calculation method and the pattern of beta diversity. So part two, Dr. Hao Minhui will introduce a new method for measuring beta diversity. And part three, Dr. Fan Chunyu will introduce the pattern of beta diversity in northeast of China. Species diversity includes three dimensions: alpha diversity, beta diversity, and gamma diversity. Among them, alpha diversity is the species diversity in a specific site or habitat. And beta diversity is the amount that connects local alpha diversity and the regional gamma diversity of a community, and quantifies the changes in species composition and structure between different sites or habitat. And gamma diversity is the total species diversity in a series of sites within a certain region. So far, there are many ways to calculate the beta diversity. For example, beta equal gamma divided by alpha bar, or gamma minus alpha bar, or 1 minus alpha bar divided by gamma. Here, alpha bar is the average of the number of species, and gamma is the total number of species in the two sites. However, regardless of how is it calculated, beta diversity is usually a simple function of alpha and gamma diversity. It means that beta is not independent of variation in either alpha or gamma diversity. So I will take the third formula, this one, as an example. As can be seen from the first picture, for the forest in northeast China, beta diversity increases with gamma diversity. And in this picture, gamma diversity values decrease with latitude. when testing the ecological drivers of beta diversity, it's necessary to correct for this gamma dependence. So a null modeling approach is needed to test if beta diversity deviate from the expected value of beta diversity based on random sampling from the species pool. Below, I will use an example to show how to do the simulation of the null model. This is example data. It includes two sites, set 1 and set 2. I will use this data to show how to calculate the degree of species variation between the two sites, that is beta diversity. There are 9 trees in set 1 with 3 species, S2, S3, and S4. And there are 6 trees in set 2 with 4 species, S1, S2, S4, and S5. Then we can calculate a species set matrix. As can be seen from this table, the corresponding number of the 5 species in set 1 are 0, 2, 4, 3, 0. In set 2 are 3, 1, 0, 1, 1. Based on this data set, the average number of species, alpha bar, is 3 plus 4 divided by 2. We can get 3.5. And gamma is the total number of species in the species pool. It's 5 here. So beta diversity value is the alpha 1 minus alpha bar divided by gamma. We can get 0.3. So the observed beta diversity value is 0.3 in this example. And I also have shown the R codes here. We can use a null model to remove the influence of gamma diversity. The species name here can be regarded as the mark of each tree. The null model is based on the random labeling of these marks across fixed location of trees. Here I will take the previous data as an example to show the realization process of this null model. The marks of individuals were randomly shuffled between two sites while preserving the total number of species, the relative abundance of each species at the location, and the total number of individuals in each site. In this way, we can get a new simulated species data, and the random labeling can be carried out by sample in our software. For the new simulated data, we can calculate a new species set matrix. In this matrix, the numbers corresponding to the 5 species in set 1 are 3, 2, 2, 2, 0. In set 2 are 0, 1, 2, 2, 1. Based on this table, the alpha bar is 4 plus 4 divided by 2. We can get 4. And the gamma is the total number of species in the species pool. It's 5. Therefore, the beta value is 1 minus alpha bar divided by gamma. We can get 0.2. In fact, this type of null model is determined by species abundance distribution. This picture shows the latitude pattern of beta diversity of forest in Northeast China. We compared the observed values of beta diversity with the expected values under the null model. Beta diversity is generally greater than expected at nearly all locations along latitude. It means that species tend to be more aggregated than expected by chance. The degree of deviation of the observed beta diversity from the expected beta diversity can be defined as beta deviation. The beta deviation can be calculated as the difference between the observed and the expected value divided by the standard deviation of randomly simulated beta diversity. This is an example to show how to calculate beta deviation. We can repeat the random labeling process 100 times, get 100 randomly simulated beta diversity values. Let's call it beta null. Then we can calculate beta mean is the average of 100 beta null values. Beta SD is the standard deviation of the 100 beta null values. Finally, beta deviation is beta observed minus beta mean divided by beta SD. We can get 1.56. This picture shows latitudinal gradients of beta deviation in Northeast China. All beta deviation values at different latitude are positive. These positive values means that higher beta diversity than expected by chance and reflect an intraspecific aggregation. As you can see from this picture, beta deviation doesn't show a monotonic relationship with latitude, but depends on underlying environment conditions. The latitude gradients of beta deviation reach a peak at 50 degrees. This part is the middle temperature zone. This part is the cold temperature zone. So this section corresponds to the transition from the middle to the cold temperature zone in the study region. Thereafter, beta deviation declines rapidly towards the cold temperature zone because of decreasing environmental variation. In fact, we found the precipitation in the study region has seen the most dramatic change. It means that the precipitation is the important driver for latitudinal pattern of beta deviation. This is all I want to talk about. Next, Dr. Hao Minghui will introduce his new method for measuring dissimilarity among communities. This method can be a very useful tool for beta diversity. Hao Minghui, can you now?
Okay. Hello, everyone.
你好，老师。
Yes, you are on.
Yeah. Hello, everyone. My name is Minghui Hao from the Beijing Forestry University. I feel honored to share my studies with you here. Sorry. Today, I want to introduce a new method for measuring dissimilarities among communities, which our method is based on the discriminating avalanche index. Traditional estimates of community dissimilarities are based on the difference in species incidence or abundance. Examples are the Yakad, Sorensen, or Gray Curtis dissimilarity index. Those indexes measure the dissimilarity among communities are based on two quantities. the number of species shared among the different communities, and the number of species unique to each community. However, we know that community dissimilarities is not only affected by species incidence or abundance, but also by the biological difference among species. Here we introduce the avalanche concept. The avalanche concept was first proposed by Ganesha in 1997, which we call this the simple avalanche here. The simple avalanche is calculated as the sum of the pairwise distance of all species within a community. Dij here represents the pairwise distance among species I and species J. It could be any quantifiable biological differences among species, such as the taxonomic, phylogenetic, and functional distances. Based on this original avalanche, we proposed a new version of the avalanche index, which we call it the discriminating avalanche. The simple avalanche was proposed to estimate the phylogenetic or functional aspect of alpha diversity within our community, while the discriminating avalanche was proposed to estimate the distance among species, among communities, which we consider this a refinement of beta diversity. Here we use a simple example to show how can we calculate community dissimilarity using the discriminating avalanche. We use species composition data from 3 forest plots in Jiaohe, northeast China. First, we need a community species abundance matrix in which the rows represent plots and the columns represent species. Then we translated the species abundance data to the species frequency data. We got the relative frequency of each species in each plot here. Then we can calculate the absolute difference of species frequency between plots. That is the delta AB in this formula. For example, for the first species, ABHO here, the plot one, the delta plot 1, plot 2 equals its frequency in plot 1 minus its frequency in plot 2. Therefore, delta ABHO plot 1, plot 2 equals to 0.007. In a similar way, for the second species, delta PIQ here, plot 1, plot 2 equals 0.03. The Dij can be the taxonomic, functional, and phylogenetic distances between species. We use the taxonomic distance here. We define the distance between two species of the same genus as 0.2. The distance between two species that belong to the same family but to different genera as 0.4, and so on. ABHO and KIPO belong to the same family but to different genera. Therefore, their distance is 0.4. Yeah. Now, based on this formula, we can calculate the pairwise distance between the 3 forest plots. This distance incorporates information not only about species frequencies, but also about the taxonomic distance among species. Therefore, this approach has advantages in discriminating among forest communities, especially when the forest composition data are collected from larger scales. Next, we use a data set that includes 35 forest plots from 15 cities and 9 countries to illustrate our approach. Each plot covers an area of 1 hectare. In each plot, all woody stems with dBH exceed 5 centimeters were recorded, measured, and stem mapped. The species composition is similar in most of the plots from our same region. In contrast, plots from different regions share few or no species. However, some of those plots may share the same genera or families, while others may differ not only in species composition, but also by genus or family. Therefore, we can use this data set to test the performance of our discriminating avalanche. This picture shows the results obtained from the discriminating avalanche. The values in each cell represent the pair distance between forest plots. The distance ranges from 0 to 1. The color from blue to red indicates the distance from small to large. To compare the performance of this new approach, we also calculate the community distance using 2 traditional methods, the widely used Bray-Curtis and Whittaker index. This is a result obtained from Bray-Curtis index, and this is the result obtained from the Whittaker index. For the Bray-Curtis and Whittaker indices, we can see that most of the pair distance are 1. This is because Bray-Curtis and Whittaker indices focus on species only. Our forest plot were collected from a global scale, and most of those plots have no shared species, and the distance between those species are 1. However, for the discriminating avalanche, there are no distance that reached a maximum distance of 1. This is because even though those plots have no shared species, they may have shared genera or families. Therefore, we can compare their dissimilarities at the genus or family levels. That's all. More information about the discriminating avalanche could be found in our publications. Thank you for your listening. Okay.
Thank you. Thank you.
Hello, everyone. Ladies and gentlemen, I am Fan Chunyu from Beijing Forestry University. Thank you very much for having the chance to speak to you all. Today, I'd like to introduce the work I'm recently involved in with Professor Zhang and my collaborator Tanling Zhao. My topic today is about the scale-dependent pattern of beta diversity and the community assembly mechanisms from local to regional. Ecologists typically measure scale in terms of extent and green. The extent of sampling area determines gamma diversity at a regional scale, and the green of sampling unit define alpha diversity at local scale. If the spatial extent of the sampling area is fixed and the spatial green is allowed to vary, then the beta diversity might decrease monotonically. This is because larger sampling green can capture a large proportion of the community, and the similarity between green units will be increased. When the spatial green is fixed and the spatial extent is allowed to vary, beta diversity will increase monotonically. This is because when new spatial green are incorporated, variation in stochastic occupancy pattern, such as the death, the recruitment, and the deterministic variation in species responses to habitat heterogeneities will result in increasing compositional dissimilarities between spatial greens. So what we want to know is, when the extent is fixed, what will happen to the beta diversity at local scale? And when the green size is fixed, what will happen to the beta diversity at the regional scale? Now we come to the community assembly mechanisms. Environmental filtering and dispersal limitation are two main mechanisms shaping the pattern of beta diversity. Environmental filtering means that environmental condition and the species niche preference can control species composition. Sampling units from similar habitats are more likely to have similar species composition. Dispersal limitation refers to the effects of spatial dispersal history. Sampling units that are located in close to each other should have a more similar species composition than units that are separated by a greater distance. It's very important to distinguish the relative roles of these two mechanisms behind the pattern of beta diversity at local and the regional scale. Let's start with the local scale. At this scale, we want to test the two following hypotheses. The first one is beta diversity of tree species decreases with increasing sampling green. The second one is the causes affecting beta diversity vary depending on the spatial green. To address these two problems, we conducted a study at 3 observational forest plots in northeastern China to detect the effects of scale on beta diversity. We divided the whole plot into different sizes based on 4 green designs, with the green size ranging from 20 multiplied 20 meters to 50 multiplied 50 meters, with increments of 10 meter multiplied 10 meter. This picture shows the flow of our analysis. The beta diversity was calculated as the total variance of community data. The details can be found in this paper published in 2013. The results shows that, as is expected, beta diversity decreased with the increasing green size. The general form of the relationships is almost identical in the 3 study areas. Variation partitioning of beta diversity was performed to decompose the variation in species composition into fractions, explained by the environmental variable and the spatial variable. The best explained beta diversity variations were found in the CBF plot, where nearly 60% of the variation can be explained by environmental and spatial variables at 40 multiply 40 meters scale. The result indicates that environmental filtering had greater effects at bigger grain sizes, as shown by the white bars in this picture. Well, dispersal limitation was found to be more important at smaller grain sizes. Our hypothesis at the regional scale is that the effects of environmental determinants on the beta gradient are strongly affected by the extent of the regional community. Our regional scale study covers a large area of natural forests in northeastern China. In the summer of 2017 and 2018, a total of 456 of 0.1 hectare circular forest plots were established. We use each of the forest plots and its nearest neighboring plots to define a particular regional community. Beta diversity was calculated using the same method at the local scale in each regional community consisting of 5 local plots and 10 local plots, respectively. This approach allowed us to assess the possible effect of spatial extent on beta diversity. The result shows that the observed beta diversity showed a strong decline with increasing latitude, irrespective of the size of the regional communities. However, the beta deviation showed a non-monotonic pattern along the latitude, and this pattern became stronger at the 10 local plots regional communities compared with the 5 plot communities. The total variation in beta deviation explained by environmental heterogeneity and latitude increased from 5 plot regional communities to the 10 plot regional communities. Measured environmental variables, as shown, the pink-colored part alone explained 30% of the variation in beta deviation for the 5 local plots regional communities. And for the 10 local plots regional communities, the environmental variables explained 58.9% of the variation. In conclusion, we found that the pattern of beta diversity is highly dependent on the sampling grain and on the extent of the regional community. Details are available for download. That's all. Thank you very much for listening.
Ban Chunyu, thanks for your presentation. Huang
Yeah, I'm here.
I see some question in chat window. 5? There are 5 questions.
Yeah.
Okay.
If you want, excuse me interrupting. I've been a little bit associated with this work, so if you want me to chair the session, I would be happy to do that. I have the questions before me. This is Klaus von Garrow speaking. I have the questions before me. And who are you, sir? Can you introduce yourself? Who was talking just now?
Yeah, Garrow.
Did you have a question? Okay, then we start with Carrie to everybody. Carrie, in the pattern of beta diversity, what is the role of the null model analysis? And Sarah asked the question that is quite similar to, I think these 2 questions were directed at Chunu. Chunu, would you like to respond to these questions?
Yeah. Okay. In fact, I have briefly mentioned this question earlier. Gamma diversity refers to the total number of species in a series of size within a certain region. It defines the size of regional species pool and determines the number of species that can enter a community. The greater the gamma diversity in the region, the more species that can be adapted and distributed in a certain habitat. And the higher the species richness in the actual community, this means that the species diversity of a community is not only related to environment conditions and ecological process, but also limited by the regional species pool. So if we want to know the environment driving forest, driving factors of beta diversity and ecological process behind it, we must first remove the influence of the gamma diversity and divide the effects poorly caused by environment factors. This process can be achieved using the Null model. In fact, so far many studies have now confirmed the importance of the Null model. It's all.
Yeah, thank you very much. I think the marks of trees is just the species. Huh? So if we randomly shuffle the marks, we randomly assign species to tree positions. That's what Sarah was asking. So then there is a question. Thank you, Chunu. Is that okay?
Oh, yeah, okay.
Then there was a question written in Chinese, and Yue Xingmin, she said, sure. So that was in the affirmative. Then there is a question about Ganeshaya's Avalanche. I think we should mention this name because Professor Ganeshaya from Bangalore, he's a taxonomist, and he actually first published this avalanche index. And the question is to measure genetic distance between the two populations, which is meaningful. How the discriminant avalanche index significantly explains the difference between the two communities. Kindly explain more explicitly. Minhui, would you like to respond to this? It's about significant difference.
Yeah, okay. Can you see my presentation?
Yes.
Yeah. Because the simple avalanche proposed by Ganeshaya, it's used to measure the alpha diversity. The DIG here represent the taxonomic, phylogenetic, of course, the genetic distance, but it's not care about the species frequency. But for the discriminating avalanche, we also consider the species, the difference in species frequencies. So it can use to measure the difference among communities, because it not only considered the species frequency, but also the distance among those species within those communities.
Yeah, thank you. I think the answer here is that this index measures the magnitude of the difference. To develop special methods for assessing a significant distance, we haven't done that yet. You know, this is a good question. I think one should take note of this, and let's go ahead with this. Okay. Thank you very much, CC8F3016, for your question. Very important question.
Professor Gardo, I am Dr. Tiwari. This is my ID.
Ah, that's Tiwari.
Yeah.
Yeah, Dr. Tiwari. We will follow this up. A very good question.
Thank you. Thank you.
So far, we can only measure the magnitude of the difference, not whether it's significant or not. And that requires more studies.
Yeah. This is my Zoom ID in my mobiles. Because
okay.
I am connected with the mobile, so I could not put my name. So I asked this question.
Thank you very much for the question.
Thank you very much. Thank you, sir. Thank you, sir.
And then there was a question from Joe. Joe, could you give us some practical suggestions? What is the best method for calculating beta diversity? Who wants to answer this?
Gardo, I can answer this question. From Joe, could you give us some present suggestion? What is the best method for calculating beta diversity? Okay. In fact, there are many methods for calculating beta diversity. And each method has its own advantages and disadvantages. It's hard to say which method is the best. I suggest you try more methods to compare and analyze, then make a comprehensive analysis based on these results. It's all. And
okay, thanks. Then they're asking, what is the main challenge in the field of beta diversity? Who wants to respond to that?
What's the main challenge in the field? Oh, okay, Gardo, I do it. It's a very big topic. But I will try. Many studies have clarified that species richness has a latitude gradient. It decreases from the equator to the poles. However, no consensus has been reached on whether beta diversity has similar latitude gradient. So the geographic pattern of beta diversity and its formation mechanism have become a hot spot in the current biodiversity research. At present, the analysis of the geographic pattern of beta diversity is mainly at the regional scale. And there are few analyses at the community level. And beta diversity is closely related to spatial scales. And beta diversity is a very important concept in ecology. It plays a significant role in testing ecological theories. One is the main challenge is identifying the cause of beta diversity patterns observed across ecological gradients, such as the global level latitudinal gradient, where recent studies generally agree that beta diversity is negatively related to latitude. Insignificant relationships have also been observed. But there is little consensus on whether community assembly process contribute to the latitude gradient of beta diversity.
Thank you very much for taking the trouble that. Well, I'm not sure if I understand everything, but whoever asked the question, are you satisfied with the answer? Would you like to ask again? Okay, then there is another question. Severity and heterogeneity of stress are major constraints of beta diversity, but their relative influence is poorly understood. How to address this question by examining the patterns of beta diversity in stress-sensitive versus stress-tolerant stream diatoms and their response to local versus regional factors along gradients of stress severity and heterogeneity. Thanks very much. We are actually concerned more with trees than with stream diatoms, but if there's Chuni, would you like to address this, or do you want me to?
Maybe Fan Chunyu can do it.
Ah, yeah, Fan. You can fall back on me, Fan, if you think it's too far out of your expertise.
I think it is, I'm not very sure about the answer, but I think we need a very comprehensive experiment to get the data and help us to get that answer.
Yeah. Okay.
I don't think it is satisfied.
Yeah. I think it's a little bit out of our focus. However, in forested landscapes and large forested landscapes, high beta diversity seems to indicate greater resilience and greater stability of the landscape as a whole. You know, if we only have a large landscape planted with eucalypts or another landscape with a mix of natural forests and other ecosystems, then the ability to respond to stress may be greater. So beta diversity is a very, very important concept, in my opinion, in trying to measure this stability. You know, we have this productivity problem, ecological productivity, but the stability, diversity, stability relationship is a very important one. And I think our group in Beijing, under the leadership of Dr. Zhang Chunu, is at the moment, I think Qiao Xuetao also, no? At the moment studying this.
Gado, still another question is from Meta, M-A-T-T. Can diversity be used for biodiversity conservation?
Yeah, biodiversity conservation.
Yeah. Okay, I will try to do it.
Excuse me, Professor Gardo, I have one query. So I was trying to relate, because in India we have large hot arid zones and we have stress severity in the forest. So whether beta, it is possible to relate the stress severity with the beta diversity, and beta diversity can explain the stress severity in arid forest. That was my question.
I'm not sure. You're talking about drought-stricken areas, dry areas?
Yeah, because I asked that question about the heterogeneity and severity, because in India we have arid forest, hot arid zone, and we have stress severity. So whether we can relate the beta diversity with the stress severity also, is it possible? That was my question actually.
Actually, I think, Dr. Tewari, maybe we should talk more about the risk. You know, the risk is another factor that is related maybe to beta diversity, that we spread the risk within a landscape. But may I come back to Matt? Chunu, you wanted to answer Matt's question. Can beta diversity be used for biodiversity conservation?
Yeah, I will do it. In fact, it's a really difficult question. The beta diversity pattern arises from two different aspects. One is the species replacement. Another one is the species loss or increase. The species replacement will lead to a decline in the number of common species among different communities, and an increase in the number of species unique to each community. While the species loss or increase will lead to difference in species richness among different communities. These two aspects together lead to difference in species composition between communities. It can be used to guide the biodiversity conservation. For example, if difference in species richness dominant beta diversity, we should protect regions with high species richness. But if species turnover dominance, we should protect a wider region. I don't know if it's right.
Thank you. Thank you. Mr. Bakar Ahmed asked or made a comment to everyone: the field in itself is challenging. I totally agree, Mr. Ahmed. That is really very complicated. There are so many different ways in which you can calculate beta diversity. There are new ways and new methods emerging all the time. So, well, there are no more questions that I can see on the screen here.
No, Gado.
Yeah.
There's another question from Sarah.
Sarah, wait. Oh yeah, Sarah at the beginning. The marks of trees were randomly shuffled between two sides.
The marks of trees were randomly shuffled between two sides. It's hard to understand.
Yeah, can you explain
more clearly? Okay, I have told a little bit about this question, but it may not be very clear. Let me explain more. The species name can be regarded as the mark of each tree. So in the calculation process, it keeps the position of the tree unchanged. And randomly matches the species name among different individuals. It simulates the process of randomly selecting species from a species pool. So it will form a forest community. This community is dominated by random process. It's all.
Sarah, are you happy? Some listeners, we cannot communicate with them. Is that so, Huanzhen? Some of the people listening, they can pose questions, but we cannot discuss.
Yeah, they are listening, and if they have other questions, I can send the question in our chat window.
Okay.
Mm-hmm.
Well, I think it's almost time to close. If there are no more questions, then we can just say that I will return to biodiversity in 2 weeks' time, trying to clarify some of the concepts. Again, it's a very important topic. Today, wow, this was a full session presented by researchers who really have been very successful, published in leading international journals. Thank you very much, Chunyu Zhang and Chunyu Fan and Minhui Hao, for your very nice presentations. And thank you everybody for their questions. So I will call it a day and say goodbye to everyone. Thank you, for
Thank you, Gado.
Hi, Stella.
Hi.
Hi.
Yeah.
Nice to see you again.
Yeah. Okay.
Yes, Lily, Stella is there. Yeah, Stella.
Yeah. Yeah. Today I could be connected.
Ah, okay. Okay.
Thank you, sir, for the very interesting session.
Oh, Dr. Madam, yes. How are you?
I was listening to you and all the very nice presentations today.
Yeah. Hey,
Happy to see Minhui, who has written that paper in vegetation science.
we will give more credit to Dr. Ganeshaya for his excellent work in 2 weeks' time. You come back in 2 weeks' time, and I will especially make an effort to highlight his work, his contribution to beta diversity.
Thank you so much.
Jaime. Yes, Jaime, yeah. I can see.
Hello.
Where are you, Jaime? In Mexico or in Germany?
I'm Göttingen.
Oh, you're in Göttingen. Oh, yeah.
Yeah.
In the lockdown.
Also, Marianne is here.
Yeah. Okay.
Yeah, I've been in Göttingen. I've been in the lockdown.
Yeah, I know. Okay, Huanzhen, you have the last word.
Mm-hmm.
You have the last